Good day. Today we'll talk about regarding streptolysin O. Streptolysin O is regarding ASO. ASO is anti-streptolysin O, meaning it is the antibody to the streptolysin, okay, toxin or streptolysin produced by the Streptococcus pyogenes. Okay, so regarding Streptococcus pyogenes, Strep pyo is a gram-positive cocci and it is usually arranged in chains. So for in cluster or arranged in cluster, that's for Staphylococcus. So for Streptococcus is the arrangement is in chain. Okay. Then the hemolytic pattern of Streptococcus pyogenes, it is beta hemolytic or beta hemolysis. So meaning there is complete hemolysis. That's for beta hemolysis, beta hemolytic pattern. And in the Lancefield grouping of Streptococcus, Streptococcus pyogenes is under Group A strep, or it is classified as Group A strep using the Lancefield grouping. Okay, grouping. Now, regarding Streptococcus pyogenes, it is the major cause of bacterial pharyngitis, okay, or sore throat, okay, and childhood impetigo. So impetigo is a skin infection. So it is common regarding strep pyogenes. The common diseases associated with strep pyo is respiratory tract infection. You have your sore throat and skin infection. Okay, example, you have impetigo. Okay, next is you have the virulence factor. Okay, so when I say virulence factor, these are substances that assist the bacteria, okay, in colonizing the host cell. Okay, so virulence factor is a substance that assists the bacteria in colonizing the um, host cell. Okay, now regarding the virulence factor of S. pyogenes, the virulence factor is M protein, the primary virulence factor. So regarding M protein inhibits phagocytosis. So how does strep pyo avoid the immune system of the host cell? By producing M protein, because M protein inhibits phagocytosis. So it avoids the engulfment, okay? So the strep pyo bacteria avoids or avoid phagocytosis by producing M protein. Okay. Next is it limits the deposition of C3. And regarding C3, C3 is a complement protein. Now, remember, complement system, the function of our complement system is to lyse the antigen. So in strep pyo infection, the antigen is the Streptococcus pyogenes bacteria. So the host will produce complement to lyse the S. pyogenes bacteria. But S. pyo is brilliant, okay? So it produces M protein that limit the deposition of C3 or limit the activation of the complement, thus avoiding lysis. Okay. Next is another function of the M protein. It helps in the attachment, okay, of strep pyo to the host cell. Okay, so it helps in the attachment of strep pyo to the host cell. Next, immunity to GAS or group A strep appears to be associated with antibodies to the M protein. And there are more than 100 serotypes of this protein, and immunity is serotype specific. So there are 100 serotypes of strep pyo, and regarding the immunity to strep pyo, it's um, serotype specific, meaning reinfection is very common. Okay, why? Because of the abundance of serotype of your S. pyogenes. So reinfection is common. That's why, for example, every year you have sore throat because you're infected with a different serotype of S. pyogenes. Okay. Next is other virulence factor. So for other virulence factor, first you have your streptolysin O. And streptolysin O uh, causes destruction of red blood cell, or it causes hemolysis. Okay, streptolysin O. The next is you have your deoxyribonuclease B. And regarding deoxyribonuclease B, it is responsible for the breakdown of DNA. Okay, breakdown of DNA. The next is hyaluronidase. So hyaluronidase is responsible for the breakdown of hyaluronic acid. So regarding hyaluronic acid, it is present in connective tissue. Now, regarding strep pyo is the cause of necrotizing fasciitis. Okay, necrotizing fasciitis. So in necrotizing fasciitis, you have fast uh, spread of the skin infection, and that can cause amputation because there will be necrosis in the infected site. So it's an emergency. Okay. So S. pyo is also known as your flesh-eating bacteria because of the capacity of S. pyo to produce hyaluronidase. Because hyaluronidase, di ba, it's responsible for the breakdown of hyaluronic acid, so meaning it facilitates the spread of the skin infection. Okay. Next is you have nicotinamide adenine dinucleotidase. So this is NADase. So NADase. So regarding NADase, it interrupts the production of ATP. Okay. The next is streptokinase. So for streptokinase, it dissolves blood clot. Okay, that's the function or action of streptokinase, to dissolve blood clot. So antibodies produced against these substances are useful in the diagnosis of infection and for the potential development of complications or sequelae associated with GAS infection. Okay. Now, regarding GAS or group A strep or your S. pyogenes, S. pyo, okay, can cause sore throat. And the problem with S. pyo, it can cause post-streptococcal sequelae. Okay. Now, regarding these post-streptococcal sequelae, it is uh, it is no example of your post-streptococcal sequelae, you have glomerulonephritis and acute rheumatic fever. So the problem with post-streptococcal sequelae, during this phase or during this period, the microorganism is already absent. Okay, it's not present in the body na. So during this time, to determine the cause of your post-streptococcal sequelae, so you need to detect or measure the antibodies against these substances. Because during post-streptococcal sequelae, you have increased antibodies against these substances. So that's how you determine if the cause of your glomerulonephritis or rheumatic fever is strep pyo because of the presence of anti-streptolysin O, anti-deoxyribonuclease B, anti-hyaluronidase or anti-NADase, and anti-streptokinase. Okay? Next, diseases caused by S. pyo, other diseases caused by S. pyo. You have pharyngitis, necrotizing fasciitis, streptococcal toxic shock syndrome. So regarding toxic shock syndrome, toxic shock occurs because of cytokine storm. So you have excessive production of cytokine. And if there's increased production of cytokine, the effect you have um, recruitment of um, immune cells to the kidneys, or example to the kidneys, and that recruitment of immune cells to the kidneys that can cause um, injury to the kidneys or lysis to some parts of the kidneys because of the lysozyme and other enzymes produced by the immune cells, and that can cause organ failure. Okay. Another is impetigo. It's a skin infection. Other complications, you have otitis media, erysipelas, cellulitis, puerperal sepsis. So regarding puerperal sepsis, it is an infection of the um, genital tract, and it usually occur after the rupture of amniotic sac. Okay, it usually occur after the rupture of the amniotic sac. Another is sinusitis, acute bacterial endocarditis, and meningitis. So these are the diseases, other diseases associated with S. pyogenes infection. Next, upper respiratory tract and skin. So the common location of S. pyo infection, it's the respiratory tract, particularly upper respiratory tract. So example, you have your pharyngitis or your sore throat. The next is skin. So you have impetigo, example. Next, streptococcal pyrogenic A and streptococcal pyrogenic C are super antigens that can induce toxic shock syndrome. And remember, if there's toxic shock syndrome, you have increased cytokine production. The next is you have the recruitment of immune cells to a particular organ. And for example, in the kidney, you have the accumulation of immune cells in the kidneys, and that can cause renal failure. Okay. Next, regarding group A streptococcal sequelae or post-strep sequelae, first is you have your acute rheumatic fever, and it's usually preceded by pharyngitis or tonsillitis. Symptoms, you have fever, pain caused by inflammation in the joints, and inflammation of the heart. Okay, so there's inflammation of the heart because regarding the M protein produced by strep pyo, it has three epitopes. And this characteristic of the M protein permits cross-reactivity. So meaning of cross-reactivity, the host immune immune system produces antibody that is against the M protein of S. pyo. Okay? But this antibody can also react with the cells, okay, of the heart. Okay? So you have the reaction of the antibody and the heart tissue. Okay? And that can cause inflammation or problem to your heart. Okay? So that's for your acute rheumatic fever. There's cross-reactivity of the antibody. Okay? That is for the M protein of your S. pyogenes. But since there is cross-reactivity, meaning it can also react with the cells present in your heart. Okay? Next, acute glomerulonephritis. So in acute glomerulonephritis, it's usually preceded by infection of either the skin or the pharynx. Symptoms, there's hematuria, proteinuria, edema, and hypertension. Okay, so these symptoms appear due to renal failure, or not renal failure directly or diretso na siya nga renal failure. There is renal impairment. Okay? So, you have hematuria, proteinuria, edema, hypertension, malay, backache, abdominal discomfort. Renal function is usually impaired, and you have immune complexes, deposition in the kidneys that stimulate the inflammatory processes or response. So you have increase or accumulation of immune cells in the kidneys, and that can cause damage to your kidneys because of the enzymes produced by the immune cells. Next, you have the laboratory diagnosis. So for laboratory diagnosis of S. pyo infection, you can perform culture, but the problem with culture, you need at least 48 to 72 hours to identify strep pyo because you need to collect the specimen. After collection, you need to inoculate and you need to wait for 24 hours. Then after 24 hours, you need to make a pure culture. Then after pure culture, that's the time you do biochemistry test or no, biochemical test to identify group A strep. So the alternative is to perform group A strep antigen testing. And for group A strep antigen testing, it only takes 2 to 30 minutes to determine whether there is group A strep present in the specimen. Next is you have your lateral flow immunochromatographic assay. So it's widely used in outpatient clinics and it's easy to perform. So the principle, first you have the reaction of the antigen, which is your group A antigen, and the enzyme labeled antibody. So if this is the sample well, your sample well contains antibody labeled with enzyme. So this antibody labeled with enzyme will react with the antigen present in the specimen. So after that, you have the formation of antigen-antibody complex. The next is you have the movement, okay, movement of the antigen-antibody complex along the test line. And in a certain point, so for the test region, this test region is coated with antibody that will capture the antigen-antibody complex. So if there's an antigen-antibody complex, the second antibody present in the test region will react with the antigen-antibody complex, thus forming or thus result in color development or line development in that test region. Okay? So if there's no antigen-antibody complex, the antibody present in that test region will not be able to react with the antibody alone. So it needs antigen-antibody complex as partner. Okay? So if antigen is absent, there's no line development in the test region. Next, antigen-antibody complex is captured by another antibody at specific location in the membrane, and culture should be performed when rapid test results are negative. Okay, so the purpose of culture is confirmation. Next is you have detection of streptococcal antibodies. So example, you have your anti-streptolysin O, anti-DNase, and anti-hyaluronidase. So microorganism is unlikely to be present in the pharynx or in the skin at the time symptoms appear. Okay, so you cannot perform culture or throat swab culture or uh, the use of other specimen in order to isolate strep pyo. So the alternative is to measure antibodies against strep or antibodies against streptococcal toxins. So the onset of clinical symptoms of rheumatic fever or glomerulonephritis typically coincides with the peak of antibody response. So acute and convalescent phase sera, if we compare this one, you have fourfold rise in the titer. Okay. And ASO titers typically increase within 1 to 2 weeks after infection and and uh, peak between 3 to 6 weeks following the initial symptoms. So next is you have your anti-DNase testing. So for anti-DNase testing, it is useful in patients of having glomerulonephritis preceded by streptococcal skin infections because AS antibodies often are not stimulated by this type of disease. And it is detected in patients with acute rheumatic fever have negative ASO test results. So the principle, you have uh, DNase reagent, okay, and the second reagent you have DNA methyl green conjugate. The characteristic of DNase, it is responsible for the breakdown of DNA, okay, and once DNase break down DNA methyl green conjugate, there's a color change from green, okay, to colorless because of the breakdown of DNA. Now, in anti-DNase test, the reagent contains DNase and the serum of patient specimen contains anti-DNase. So the DNase in the presence of anti-DNase will be neutralized. So meaning no available DNase to cause breakdown of DNA methyl green conjugate. So meaning there's no color change if anti-DNase is present in patient specimen. But if anti-DNase is absent in the patient specimen, the DNase will be available to break down DNA methyl green, thus producing color change. So from green to colorless. Okay? So In this procedure, you need serial dilution. So for example, the first tube we have 1 is to 2, second tube we have 1 is to 4, third tube we have 1 is to 8. So for example, for the first tube we have color change, no color change, meaning the DNase was neutralized by anti-DNase present in the patient specimen. Then for tube number 2, you have color change from green to colorless. Okay, so meaning anti-DNase is absent, thus uh, anti-DNase is absent. So the effect, you have DNase available to break down DNA. So the effect, you have the breakdown of DNA methyl green. So you have color change from green to colorless. Now, in order to identify the titer, the reciprocal of the highest dilution demonstrating a color change intensity of between 2 plus and 4 plus, meaning no color change. The titer is the highest dilution with no color change. That's for the titer. So meaning this tube is meaning there's no enough anti-DNase. So the titer for this in this situation it is 2 because tube number 1 demonstrate no color change, but tube number 2 demonstrate color change. Okay, so this one is more of semi-quantitative. So if we want proper quantitation or a quantitative test, then we need to perform nephelometry. So for nephelometry, no need to observe color change because our goal in nephelometry is to measure the amount of scattered light. Okay, and the amount of scattered light is directly proportional to the concentration of anti-DNase. Okay. Now, regarding ASO determination or anti-streptolysin O, ASO is the antibody to streptolysin O. Okay, and it indicates the recent streptococcal infection in patients suspected of having post-streptococcal sequelae. Okay, and the traditional way of determining ASO titer or ASO is the use of streptolysin O as the reagent and um, RBC because streptolysin streptolysin um, causes hemolysis of red blood cells. Okay, so for this or in this traditional method, the reagent contains streptolysin O, which is capable of destroying red blood cell, the second reagent, and we're using serum as the source of ASO, or the patient specimen is serum, or the specimen is serum that possibly contains ASO. Okay. Now, for the principle, if ASO is present in patient serum, this ASO will be able to neutralize streptolysin O. So after incubation, then you add RBC. RBC will not be hemolyzed. Why? Because streptolysin O was neutralized by ASO. Okay. Now, what if ASO is absent? Okay, in patient specimen. So meaning streptolysin O is available to cause hemolysis to red blood cell. Okay, so hemolysis, streptolysin O was not neutralized by ASO. Titer was reported as the reciprocal of the highest dilution demonstrating hemolysis. So for example, you have tubes of serial dilution. So 1 is to 2, 1 is to 4, and 1 is to 8. So for example, in this tube, you have um, no hemolysis, hemolysis, and hemolysis. So correction, titer was reported as the reciprocal of the highest dilution demonstrating no hemolysis. Okay. So in tube number 1, you have no hemolysis because ASO is present and ASO neutralized streptolysin O. Then since streptolysin O was neutralized, uh, no, streptolysin O will not be able to cause RBC hemolysis. So that's why the result is no hemolysis. Tube number 2, hemolysis, meaning ASO is absent. So streptolysin O is present or available to destroy the red blood cell. Same with test tube number 3. So the titer for this one is this one, the reciprocal of the highest dilution demonstrating no hemolysis. So it is 2. Okay. So for adults, the significant titer it's 240 TOD units, and for children it's 320. Okay. The problem with this method, it's labor intensive, so the alternative is to perform nephelometry. Okay, so same with your anti-DNAs, same principle. You have the antigen-antibody complex, and that can cause scattering of light. So in nephelometry, we measure the amount of scattered light, and that is directly proportional to the ASO concentration. So ASO titers typically increase within 1 to 2 weeks after infection and peak 3 to 6 weeks following the initial symptom. Now, another way of measuring ASO is using latex. So latex agglutination. So it's simpler and uh, easy to perform. So in latex agglutination, the latex particle is coated with streptolysin O. So if this is the latex particle, it is coated with streptolysin O. So if the patient specimen contains ASO, so the ASO present patient specimen will react with streptolysin O coated in the latex particle, coated on the uh, latex particle. And after that one, after the reaction, you have the antigen antibody complex formation. And it will give you, or it will show agglutination. Okay, so if there's agglutination, meaning the result is positive for ASO. And you can also perform serial dilution to this one to measure the titer. Okay, next test to detect streptococcal infection, you have streptozyme testing. And in streptozyme testing, you're using 5 antigens to react with 5 antibodies, possibly 5 antibodies. Okay. So streptozyme test is positive in 95% of patients with acute glomerulonephritis. So in this one, you're using SHIP RBC coated with streptolysin O, streptokinase, hyaluronidase DNase, and NADase. So you have your RBC coated with 5 antigens. So the 5 antigens are um, streptolysin O, streptokinase, hyaluronidase, DNase, and NADase. So the purpose of this one is to detect 5 antibodies. So for example, if the patient's specimen contains ASO, the ASO will react with the SO. Okay. Now, what if the patient specimen contains anti-hyaluronidase? So this anti-hyaluronidase will react with hyaluronidase. Okay. Same with other antibodies. So there is hemagglutination if there's a reaction between the antibody and the antigen present on the surface of the SHIP RBC. Okay, so hemagglutination represents a positive test. Okay, next is you have your rheumatoid factor. So for rheumatoid factor, it is associated or seen in rheumatoid arthritis. So it is used to help in the diagnosis of rheumatoid arthritis. Now, what is rheumatoid arthritis? So it is an autoimmune disorder, so meaning there is the production of autoantibodies, and women are 3 times as likely to be affected as men, and it is more prevalent in women who are more than 65 years of age. Characteristic, there is chronic, symmetric, and erosive arthritis. Okay, chronic, symmetric, and erosive arthritis. And there is a decline in the functional mobility, okay, due to the inflammation or damage to the joints. And there's also reduced life expectancy because individuals with rheumatoid arthritis are prone to develop cardiovascular disease, okay, prone to develop cardiovascular disease. Next, the gene that codes for the production of that codes for rheumatoid arthritis, it's HLA-DRB1 gene. And in rheumatoid arthritis, patients with rheumatoid arthritis are usually positive for rheumatoid factor or anti-CCP. Okay? So meaning of anti-CCP is anti-cyclic citrullinated protein. Okay? Anti-cyclic citrullinated protein. And the strongest risk factor of rheumatoid arthritis, it is cigarette smoking. Okay? Next, so this is the pathophysiology for your rheumatoid arthritis. So first is you have the development of antibodies. You have your RF and anti-CCP. And RF will react with the FC portion of your IgG. And if there's a reaction between RF and IgG, then you have immune complex formation. So let's say if this is the bone, then you have the cartilage, then bone, then you have cartilage, and this is the synovial membrane, then synovial membrane. So this is our joint. Okay? So muna siya atong joint. Now, in rheumatoid arthritis, you have the production of rheumatoid factor. So RF is also present in the synovial fluid. Now, if that synovial fluid contains RF, the RF will react with IgG. And if there's a reaction between the RF and IgG, you have immune complex formation. And once there is immune complex formation, the next step is you have the activation of the immune system. And once the immune system is activated, you have cytokine production. Okay? So if there's cytokine production, next step is you have fibroblasts-like synoviocytes or increased production of fibroblasts-like synoviocytes in the synovial membrane. So you have increased production of fibroblasts-like synoviocytes. Okay? Now, the problem with these fibroblasts-like synoviocytes, it produces proteases. Okay? It produces, or fibroblasts-like synoviocytes produce proteases, and the function of proteases is to digest the cartilage. So, the protease produced by fibroblasts-like synoviocytes will try to digest or cause damage to the cartilage, or it digests the cartilage, thus making the joint painful, or movement of the, or ang pag-use sa imong joints or movement, if there's movement, it is painful because the cartilage that covers the end of the bone was digested, or natay thinning of the cartilage. So once mag rub, or there's friction between bone to bone, it's painful because of the absence of the cartilage. Okay? Another cause, or another effect of fibroblasts-like synoviocytes, you have the increased production of RANKL. So RANKL is receptor activator of nuclear factor kappa B ligand. Receptor activator of nuclear factor kappa B ligand. So regarding RANKL being that is produced by the synovial membrane, no, the fibroblast-like synoviocytes in the synovial membrane, the rank L promotes, okay, it promotes osteoclast activity. Okay, so you have increased osteoclast activity, so sa bone, so you have digestion or breakdown sa imong bone, so there is bone erosion. Okay, so that's why there's bone erosion in uh, rheumatoid arthritis. That's why it's classified as erosive arthritis. There is bone erosion due to the production or increased osteoclast activity. Now, another cause of rheumatoid arthritis is the production of anti-CCP. And anti-CCP reacts with citrulliнаted proteins present in the joint, and it causes immune complex formation. Then same pattern or flow, you have activation of the immune system. The next is cytokine production. Increased cytokine production, increased formation or proliferation of fibroblasts-like synoviocytes. And the fibroblasts-like synoviocytes produce proteases that digest cartilage and rank L that promotes osteoclast activity, thus resulting to bone erosion. Okay. Another, regarding fibroblasts-like synoviocytes, the fibroblasts-like synoviocytes usually travel from one joint to the other. Okay. So for example, if there is rheumatoid arthritis in the right hand, the fibroblast-like synoviocytes tend to travel to the left hand. Okay, so the joints in the left hand. So meaning in rheumatoid arthritis there is symmetric arthritis, chronic symmetric arthritis. Symmetric meaning both sides, the right hand and the left hand, kay affected siya or there is arthritis. Okay, so it is chronic, symmetric. So both uh, the right and left side kay affected. And it is an erosive arthritis because there is bone erosion. Okay. Clinical signs and symptoms. Initial symptoms of RA involve the joints, tendons, and bursae. Joints are typically affected in a symmetric fashion because of the movement of fibroblast-like synoviocytes. Joint and stiffness are usually present in the morning and improve during the day. And the disease can progress to larger joints like the knees, hips, elbows, shoulders, and the cervical spine. The next, if left untreated, it can result to permanent joint dysfunction and deformity. And osteoporosis occurs about 20 to 30% of RA patients. Then development of secondary disease, it's typical. You have development of Sjogren's syndrome and Felty syndrome. So for Sjogren's syndrome, you have dry eyes and mouth. For Felty syndrome, it's a combination of rheumatoid arthritis and neutropenia plus splenomegaly. And the most common cause of death in RA patients is cardiovascular disease because there is inflammation, and inflammation promotes atherosclerosis. Next, laboratory diagnosis, clinical manifestation. So to diagnose RF, you need to diagnose RA or rheumatoid arthritis, you need clinical manifestation, radiographic findings or imaging test, and laboratory testing. So for initial diagnosis, you need to check for RF, so or rheumatoid factors. Rheumatoid factor are IgM, and it reacts with FC portion of the IgG. Now, 70 to 90% of patients with RA test positive for RF, so not all with RA will have positive RF results, and it's present in about 5% of healthy individuals and 10 to 25% of those over the age of 65. So there are healthy individuals with RF. Okay. Next, diseases with RF. You have your rheumatoid arthritis, then Sjogren's syndrome, scleroderma, mixed connective tissue disease, and chronic infection. So in rheumatoid factor, we usually use latex or charcoal as carrier. So it's latex agglutination. But the problem with this one, it only detect IgM. So in latex agglutination reaction, you have uh, latex coated with um, IgG, okay, the antibody. So let me read that one. So the RF latex test is rapid slide agglutination test for direct detection and semi-quantitation aromatic factor in serum. The antigen, which is a particulate latex suspension coated with human gamma globulin. Okay, so the latex particle is coated with antibody. Okay, and the patient's serum possibly contains uh, RF. So the RF will react with the immunoglobulin attached to the latex particle or the carrier. And if there's a reaction between the RF and IgG, you have latex agglutination, or if there is agglutination. Okay. Next, anti-CCP. So for anti-CCP, this is your anti-cyclic citrullinated protein. So it's usually detected or measured using ELISA, and 20 to 30% of RF negative patients are positive for anti-CCP. So there are um, patients with RA that will have negative RF result. Now, the better marker for RA, it is your anti-CCP because it's more specific than RF. And if we combine RF and anti-CCP, it will increase the specificity of the testing to 98 to 100%. Okay. So regarding RF, it's not that specific because it's also increase in Sjogren's and other conditions. Okay. Chronic disease, chronic infection, there's increase in RF. So to increase the specificity that uh, to increase the specificity of RF, we need to also check the concentration of anti-CCP. So positive RF, positive anti-CCP, that is 98 to 100% specific for rheumatoid arthritis. Okay, so I think that's all regarding ASO and RF. Thank you.